Can you hear me? 38.3 million seconds. That makes 633,600 minutes, or easily said, 14.5 months that we have now been waiting for this moment to come. Welcome to the presentation of the first team from the ESBS Strasbourg. As you might have already seen, our t-shirts were a little bit tighter than we expected, so well, after Randy Redberg's promotion tour in September 2007 in France, we created our own team consisting of 7 students. That means our team is totally student-run and organized. It consists of our 2 nice girls, Katja and Sandra, Michael, Paul, Robin, and Manuel, and me. For those who might be not familiar with Strasbourg, I hope you are. Strasbourg is said to be the capital of Europe, and it's located in the center of Europe. It's in France, next to the border to Germany and to Switzerland. And in this spirit, also the school is a joint venture of 4 universities in these 3 countries. Namely, speaking of University in Strasbourg, University in Basel in Switzerland, and the 2 participating German universities, Freiburg and Karlsruhe. Thus, one could say the team's nationality is European. All teams had a lot of exhaustive brainstorming session and journal clubs, but we got really excited about one idea in particular. And then after we started and proceeded for several months, we accidentally stumbled upon this presentation of Drew Andy and about the implication of synthetic biology. And there he said that one of the most potential tools that synthetic biology could offer would be a device that could count. And thus leads us to our project, the first step toward an eukaryotic cell division counter. To construct our system, we decided on using binary counting, which makes it easily extendable with further bits. For example, here we start with just a normal cell, which has no color. But for the first bit, you see even a change. And for this example, green color with CFP. And then it would be a switch again to a normal color. But if you would extend it to another bit, you could maybe say blue. Then you would have 0, 0, 1, 1, 0, and those both together as 1, 1. The first step is, though, to implement a cell cycle dependent toggle switch. The optimal organism to use for this approach would also be the model organism for eukaryotic cells, which is yeast, namely S. cerevisiae. The only drawbacks we had were that there were only very, very few biobricks annotated to this moment. The fields one could imagine are quite huge and large, but especially in the field of medicine or health, here is the detection of abnormal growth. And this means, for example, that one could observe proliferation in cancer cells as spatial variety occurs. And Michael will now go more into detail in the insights of our idea.
Okay, thank you, Marius. First of all, I want to stress that what I'm presenting now is more or less a toggle switch. So, in fact, it is a 1 bit. But in the way how we constructed our modules, it is very easily extendable, so that we could not only count to 1, but to higher numbers like 3, 7, 15, and so on. So, our system. Here you can see a scheme of the cell, and you see our black box containing our system. You have an external signal, which could be any signal you could imagine. But for our purposes in yeast, it was the galactose-glucose shift, which is a quite common way to express a gene and afterwards shut it off immediately. And on the right side, you see the outcome, what should occur. It's a pattern of 1, 0, 1, 0, 1, 0, like a binary counting. So now let's have a look what is in our black box. There's first of all an inducer which recognizes the external signal and starts with counting. And then there are two modules, the 0 module and the 1 module, which architecture is identical. They only differ in binding sites, DNA binding sites, and promoters. And those two modules interact with each other. And we'll now have a look what exactly is in one of these modules. So first of all, we need a self-activator. This is important to keep our system stable at every point of time and to control it. Yeah. And then we have a repressor of the other system, and we have an activator of the other system. Those two together result in repressing of the other system. But at a certain point of time, we also want to have a switch. This is why we need the activator. Now, how do we relate our system to the cell cycle? Here, the most important is the APC, the anaphase promoting complex. This system occurs naturally in yeast and is responsible to degrade proteins during mitosis. And we just have taken these tags responsible for the degradation and put it on two of our proteins, so the self-activator and the repressor. So now let's have a look how our system works. First of all, the inducer is induced, and it uses our counting system. Therefore, a biomarker is expressed here. It's a GFP. Then, during the whole cell cycle, this expression is stable. But then the cell enters to mitosis. The APC tag is becoming active and is degrading our self-activator and our repressor. And the only one which is resting is a cross-activator. And therefore, our system switches. The color disappears. And as you can see, the other module is exactly built up like the first module and reacts in the same manner. So they have self-activation, repression, and activation of the other system resulting in repression. And the cycle re-begins. But up to now, they are only talking about theory. And a lot of assumptions have been done by us. And that's why modeling became so an important part to us. And that's what Robin has done. And he will talk to you about this.
Thank you, Michael. Well, first thing I want to mention is that modeling has been at the very basis of the systems construction for us. So we did all the characterization of the single parts we used while we did them through modeling. So there have been several questions raised. And well, first of all, here you can see the Simulink presentation of our model. And the first question to be solved was about protein half-life time. So is the dilution naturally occurring? Is that sufficient? Or do we need a further degradation motif to get rid of the pattern, of the expression pattern of the previous cell cycle? Another question was about the nature of the APC signal itself. So there are different choices in terms of duration and of transferred half-life time. And most importantly was the question about promoter construction. So questions like the expression ratio of self-activator, cross-activator, and repressor. As we heard, there's an expression of repressor and cross-activator at the same time, what has to result in repression. So it's crucial that there is a different strength in expression. Then the question of number of operator repetitions. We're talking about a switch here, and so we're very interested in cooperativity. And the more operators we have, the more cooperativity we can observe. And there's the question of shall we use the same DNA binding domain for activator and repressor, and though also the same operator, or shall we use two distinct operators and so also two distinct DNA binding domains? On that question, I want to focus in my next slide. We called the two systems the one operator system and the two operator system. In the one operator system, we only have one operator, and we use same DNA binding domains for activator and repressor. Whereas in the two operator system, we have distinct DNA binding domains, here in green for activator and in red for repressor, two different ones. How can we express that in terms of equation? The basic equation we used was the Hill-Kind equation for expression, but there is only the activation or the repressive effect. But now we have activation and repression at the same time. And for the one operator system, we imagined just to take a plain DNA binding domain as repressor, and so that would be the equation for competitive inhibition adapted to the Hill equation. And with different ratios of activator to repressor expressed already at a ratio from 1 to 2, we can observe less than 10% expression. For the two operator system, it was already more difficult. But we could imagine that that is like a repressed system where the term for maximal expression is exchanged by the activation equation. So it's the two formulas put one into the other. And here, already at lower activator to repressor concentrations, as here 1 to 1 for green, well, what is not very clearly visible, it's down here, we have far less than 10% expression. The drawback of that two operator system is that it's not really the ratio that decides if the system is repressed or not. As soon as we have a certain amount of repressor, the system will be repressed. So what we want is that it's more dependent of the ratio. That's why we preferred the one operator system, but there the effect is not strong enough. So we wanted to couple an additional repression domain to our DNA binding domain. But so we have the active and the passive effect of repression. The passive effect through competition, the active effect through the repressor. And we had to find another equation, and that is derived from the saturation equation of allosteric enzymes. And what does it look like? That would be the expression pattern with, in blue, the 0 expression, and in green, the 1 expression. And well, we can see here, during the first generation, we only have 0 specific expression and no 1 specific expression. And with the next generation, that pattern changes, and we have only 1 specific expression and no 0 specific expression. In terms of transcription factor concentrations, that looks quite similar. Here in blue, we have the repressor 0, and in green, we have the repressor 1. And, well, we can also see the switch. How we get up with the expression pattern, and through the APC input, we have the faster degradation, and the system switches. That is theory, and in theory, it seems to work fine. But now Manuel will tell you some details about the practical part.
Thank you, Robin. Well, the question is, how did we realize this? Also, in respect to this, what Robin was saying about the modeling right now. The first important thing is that the two devices here are expressed on the same level. Secondly, it's important, Michael pointed this out, and Robin as well in the modeling part, that the cross-activator and the repressor are on a certain ratio level, for that we have the inactivation when the two of them are there. We reached this goal with a modular promoter system. This is in blue. You can see several operator copies which have specific binding affinity for example, LEX-A binding domain. By coupling several of these are about 20 base pair sequences, by coupling several of these, we can we can reach a cooperative effect, and thus we can regulate the level of the expression of the two. For the expression of the cross-activator and the repressor, we have different mutated SIC1 promoter, and which are bound before the sequence here, which reach from 10 to 100% of the wild type SAC1. Okay, secondly, how does these parts look like in detail? Well, we've already said they are very similar. For that reason, we look at a general scheme right now. So what we have is a binding domain, it's binding to DNA, an activation domain, respectively a repression domain, depending whether we talk of an activator or a repressor. Then we have an X of P because we want to follow the path of our protein. And finally, the tags. The NLS tags, which allows the protein to go into the nucleus. The APC tag, which is crucial for our project, that the degradation only occurs in a certain time window. And the PEST sequence, which allow us to establish a certain protein half-life time. It is important to mention that all these parts here, or all these domains, are biobricks. For that reason, it's very important that we can make infusion, in-frame fusion proteins. Therefore, we read the Phillips and Silver technical note 2006, and we cut off the two bases upstream and downstream of the normal biobrick. For that, we can assemble two biobricks, which give rise to an in-frame assembled biobrick with just 6 bases in between. Okay, here I just want to point out what activation domain we use, the VP16, to activate polymerases. From cerevisiae, we took the repression domain TUP1, and binding domains, we established several, TED-R, C1, LAC-E, LAX-A, and GAL4, and the APC tag. Well, we developed a measurement scheme, an exhaustive measurement scheme, which should clarify further this question. What is the optimal arrangement of domains? What strength of the promoters do we need? How often do we have to put the operator in chain? And also, we have to verify the functioning of the tags. This is one of these measurements, the optimal arrangement of the domains. This is what we assembled in the laboratory, beside others. Some of them are 20 biobricks in chain. And unfortunately, we did not have the time to verify them into yeast. So they're waiting for us at home in Strasbourg. Okay, therefore, the main contribution of our team to the IGEM competition and the synthetic biology community are 27 new parts we submitted. 13 of these parts are completely new to the registry. They have never seen this sequence. The most important, the most important of them are VP16, TUP1, and a whole subset of operator sequences which allowed the fine-tuning of the expression levels. Finally, we want to thank our sponsors from the industry, from our partner universities, but also the Embassy of France here in the US. Also, we were a student-run team. We had a lot of support from the ESBS, also from the other universities. We want to thank these guys, and we want to thank you also for your attention, and are open for questions right now.